I wouldn't do anything. Send it over.
So hello and welcome to our webinar. This is a webinar that's co-sponsored by SwitchGear Genomics and by Molecular Devices. I'm Kathy Olson, Application Scientist at Molecular Devices, and I'll be kind of your host for the webinar. Today we have a featured speaker, Gregory Cooper from HudsonAlpha Institute for Biotechnology, and he's going to be discussing the discovery and functional fine mapping of expression quantitative trait loci in primary liver tissue. So key parts of this research were enabled by the SwitchGear Genomics technology that's going to be introduced by Rini Saxena. And in the last part of the webinar, I will introduce the SpectraMax L Microplate reader that was used to generate some of the data. So here is our agenda for today. So we'll start off with the introduction to the SwitchGear Reporter Assay Technology and Services by Rini from SwitchGear Genomics. And then we'll move into the main science body of the talk, which is going to be presented by Gregory Cooper. And then we'll finish up with my introduction to some of the instrumentation. So with that, I would like to go ahead and pass the presentation over to Rini so that she can begin presenting her slides. And Rini, you should have the ball now.
Yes, I am getting that ready.
Okay.
My name is Rini Saxena. I'm from SwitchGear Genomics. And today I will be discussing SwitchGear's full reporter assay solution for gene regulation studies. But I did want to start with our service offerings. So SwitchGear offers several reporter assay services, including our pathway screening service, in which we measure the effects of compounds on a variety of biologically relevant pathways using our experimentally validated promoter reporter assays. And then secondly, we also offer a microRNA target validation service, in which we use our validated 3' UTR reporters. And then lastly, today I'll be discussing our sequence variant analysis service, in which Dr. Greg Cooper utilized to gather data for his study that he will be presenting shortly. Our sequence variant assay service allows researchers to understand the functional impact of sequence variation on promoters and 3' UTRs. So here I'm explaining the workflow of our sequence variant services. And I'll take you through the steps here. So first, you would be identifying the promoter's 3' UTRs or long-range elements of your choice. Then secondly, you would specify the variants you would want us to create, whether they be individual SNPs or mutated motifs. And we also do analyze larger haplotypes from individual genomic samples. The third step is you would choose from one of the cell lines listed here, CLA HT1080, HCT116, or HepG2. We would then perform the transfections and read out luciferase activity on the molecular devices SpectraMax L Luminometer. And then lastly, we would send you your data, which would include a complete raw data set and the statistical analysis of significant differences between variants. And so to give you a brief overview of not just the services that we offer, but the breadth of the SwitchGear reporter assay solution, we do offer a collection of 18,000 endogenous human promoters, and that can be purchased off the shelf. They're ready to transfect. Similarly, we also offer 12,000 endogenous human 3' UTRs. Again, these are also ready to transfect. They're pre-cloned. We do offer, with our full solution, optimized transfection and luciferase assay reagents that have been optimized with all of our clones. And I did mention, of course, the services on the previous slide, where we do offer high-throughput target screening and variant analysis. And then lastly, we do have validated biomarker sets, which consist of several product lines, including validated assays for hypoxia, nuclear receptors, CREB, NSKB, P53, and among others. We also offer validated microRNA target reporters. Again, those are validated through prime UTR plasmids that have been validated against specific microRNAs. And then lastly, we do have synthetic reporters, which contain synthetic response elements. And that's for both studying microRNAs as well as synthetic promoter elements. So with that, I would like to introduce Greg Cooper from HudsonAlpha, who will be giving, again, the bulk of the scientific presentation.
Okay, thanks, Rini. So I'm going to pass over presentation to Greg now. And in just a moment, Greg, you'll be able to put up your slides. Okay?
Okay. Give that a second to pop up here. Hi, okay, so like I said, my name is Greg Cooper. I am a faculty member of the HudsonAlpha Institute, although most of this work was performed while I was a postdoc in Seattle at the University of Washington, working with Mark Reeder, who's a faculty member there. So the big picture for me, and I think for lots of individuals, is using genomic techniques to find causal genetic variants. So these are variants that have a functional effect that influences disease risk or normal phenotypic variation. And one of the major challenges that we all face is that genetics alone is often insufficient to specifically pinpoint causal variants. And one ironic downside to doing genomics is that genome-wide experiments get rid of prior hypotheses. Basically, you no longer have to have, or at least pretend to have, specific knowledge ahead of time about which genes are important. Instead, you just test all of them. But the downside is that when you have this sort of baseline naive hypothesis, that you result in a large multiple testing cost, which basically means it's statistically difficult to distinguish true from false correlations. And what I'll talk about today, and the sort of major motivation for this work, is that prior knowledge for different individual genes and individual variants is in fact available systematically and can be used to generate better hypotheses while still in the context of a genome-wide experiment. And so one example of this is to look at function. So if we could somehow isolate variants in the genome that were functionally relevant, then we could, in principle, boost power to discover those variants that are disease relevant. And one classic example of this is to look at non-synonymous variation. So this is basically just leveraging the sort of fact that, or assumption, which is turned out to be true over and over again, that variants that affect protein sequence are heavily enriched for variants that cause disease or variants that change your disease risk. However, this functional classification is somewhat unsatisfying in the sense that it really ignores roughly 99% of the genome, more than 99% effectively of all common genetic variants in particular, are not in fact non-synonymous. And so we're leaving large chunks of genetic discoveries off the table if you only focus on non-synonymous variants. So today we'll be talking about one way to get at that other 99%, which is to look at expression QTL mapping, or expression quantitative trait locus mapping. So the basic idea here is that variation in gene expression is a highly heritable trait. So for many genes, individuals vary quite a bit in terms of the level of expression of that particular gene in any given cell type. But that level of variation, that difference between individuals is heritable. So offspring of parents are more similar to parents than they would be to randomly selected individuals. So and in many cases, we know that variance in gene expression is a functional contributor to change in physiology or disease risk. So the connection here is that if we can find genetic variants that contribute to differences in gene expression, which in some ways, and I'll show, are easier to identify than genetic variants that directly associate with complex traits, then we can sort of use this expression as an intermediate trait to identify those variants that are functionally relevant to disease. And so the question then becomes, how do we identify causal EQTNs? And so instead of QTLs here, we're talking about QTNs because we're really interested in drilling down to individual nucleotides, so expression quantitative trait nucleotides here, so if we can pinpoint specifically causal functional variants. So the basic paradigm here is to perform genome-wide expression profiling using arrays, genome-wide genotyping, again also using arrays, And the goal then is to take those expression measurements, correlate them with SNP genotypes among a set of samples, and then identify SNP gene pairs that are highly correlated, strongly correlated. And then once we have that list of pairs of genes and SNPs, to go through then and figure out, can we find evidence for individual SNPs that appear to be functionally relevant to changing an expression level. So the basic data set we're looking at here is we were interested in primary tissue, so there's advantage of doing primary tissue. You avoid sort of potential artifacts associated with cell line immortalization. And we focused on liver in part because this was originally conceived of as a resource for pharmacogenetics, but also livers have advantage to interpreting other traits such as metabolic traits, cholesterol, that sort of thing. So the data sets are sort of two main data sets I'll talk about include 60 livers that were assembled or collected at the University of Washington and analyzed there in Debbie Nickerson and Mark Reeder's labs. And these were genotyped on the Illumina 550K array, so this is 500,000 SNPs or so. And expression was assayed on Illumina's Ref8 expression chip. So this is about 22,000 genes that we get expression measurements based on hybridization and fluorescence. We also had, and this is a collaboration with a colleague, Christopher Brown, who is at the University of Chicago and works with Kevin White. These were 220 livers that were genotyped on a similar product, the Illumina Quad 610 chip, which is basically you can downsample to the 550K content. Christopher's livers were assayed for expression on adjuvant 40K chips, which target about 40,000 genes, and basically only focused on genes that we could constantly sort of intersect between the Illumina and adjuvant chips here. And then we compared this to previously published data that had been generated by Merck on about 250 livers. And I won't get into it in detail, but we used software from Matthew Stevens' group at the University of Chicago called BIMBAM, which which allows us to both do imputation, which means that given the directly genotype variants, we can predict or impute the variants that are analyzed as part of the HapMap on our samples just by basing those guesses on known patterns of linkage to equilibrium. And BIMBAM also does what's called Bayesian regression. So this is a slightly different way to measure the strength of evidence for association between a given SNP and a given expression level. And this produces a value that's called a Bayes factor, which is basically a log-scaled measure of the strength of association between a SNP and a gene. So just to give one example here, on the left-hand side of this plot, I'm showing you the EQTL evidence for a gene called PRMT6. It's located on chromosome 1, as you can see on the x-axis here. What I'm showing you is for each SNP that was genotyped plus imputed, so you're looking at directly genotyped SNPs and those that could be imputed from the genotyped SNPs. And I'm showing you the strength of evidence of association between each of those SNPs as these open circles and the expression level of PRMT6. So you can see in background levels to the left and to the right of this plot, we get very weak evidence or no evidence, scores that hover around near 0. But as we get close to the gene here, which is this red bar indicates the extent of the PRMT6 transcript in the genome, you can see that we identify a number of SNPs that are strongly correlated with expression, and these go up to Bayes factors on the order of 20. Keep in mind that this is a log 10 scale number, essentially with 0 indicating no evidence, and typically values of 5 is where you start to get interested. You can see we have very, very strong evidence for correlation here. And if we look at the best individual SNP here, RS2335077, we can actually look at that and see what that relationship looks like for that particular SNP, which is on the right-hand side. Here we're showing you normalized expression levels. And this is in the 220 Chicago livers. And we can see the 3 genotype categories here, GG, GA, and AA. And you can see that there's a very strong linear additive effect associated with a reduction in expression level associated with the A allele at this particular SNP. So this just gives you an idea for what an individual EQTL looks like, and we'll revisit this example later. So aggregating over the entire genome, simply asking how many EQTLs, how many gene SNP pairs can we identify that are significant. In fact, we identify hundreds, and in in this plot I'm showing you it's a sort of Manhattan-style plot where I'm showing you for each gene along the genome here, and the chromosomes are labeled in alternate colors, so chromosome 1, chromosome 2, etc., alternating black and gray. We're showing you for each one of those genes the strongest signal of association for essentially all SNPs in the genome. So we essentially treated each gene as a trait, and we asked for, does it associate with any SNP anywhere in the genome? And I'll show the data later, but by and large they are all cis effects, which means that the best SNP is usually close to the gene itself. And you can see here that we identify lots of very strong correlations. So again, if we look at this Bayes factor measurement here, most of the best gene SNP pairs that are essentially random, this is essentially noise, are close to 0. But many of these range up to 10 or 15 or even in the 20 and 30 range. And individual genes, when they replicate, when a strong effect is identified in the Chicago livers and it replicates in the Seattle livers, which are on the bottom here, that gene is labeled, and then we just prune the labels to make it somewhat legible, so you can see individual gene examples. And the difference in scaling here, the y-axis on UW versus Chicago, this is effectively a difference in power because there's only 60 samples down here and there's over 200 up here. So some of this lack of replication and or the shift in scaling here is due to a differential in power. But nevertheless, we identified lots of eQTLs spread across the genome, and many of which reproduce. And one thing that this is sort of, again, getting back to the original motivation, is that you're hoping to use these data to make some more sense and help identify causal connections between expression levels and complex disease or complex traits. So here's an example of when we compared our list of SNPs that correlate, SNPs that are eQTL associated with SNPs that are associated with various disease endpoints. So these data were assembled from the NHGRI database of GWAS hits. And what we did was we asked for all those SNPs that associate with expression, identify any SNP that is in the GWAS database that correlates strongly with that, so in this case an R-squared of 80%. And we identified many, these are essentially hypothetical but reasonable hypotheses about connections between expression variants and phenotypes. So one example I'll point out here is this locus, this gene called sortillin-1, that in this right-hand column you can see whether it's replicated in one or both studies. So there was actually 3 studies. There was the Chicago, the UW, and the Merck samples. So this is a doubly replicated association. So we saw this in all 3 liver panels. In the Chicago data, it had a very strong Bayes factor, about 24. And this, in fact, this variant near sortillin-1 also correlates strongly with heart disease and LDL cholesterol levels. And we'll talk about that a little bit later. And another example here is warfarin maintenance dose with this gene called VKORC1. This was originally identified using the University of Washington livers by Mark Reeder because it associates with, this is a common variant that explains a large fraction of variation in warfarin dose. So you can see that we're starting to make these connections, and this is a theme that's repeated by various other groups as well, between eQTL SNPs and complex disease-associated SNPs. However, what we're still interested in is finding specific variants. So those still represent just correlations, and what you'd really like to know is, can you identify functional variants? Well, one pattern that's relevant to emerge is that when you look at the distribution of the best SNP associations with any given gene, you get a very sharp enrichment near the transcriptional start site or near the promoter. And a similar, not quite as striking signal is observed towards the 3'-TR, but I'm just showing here the promoter data. So here, for these curves here, showing you the count of strong eQTLs that we observe. In the black are all eQTLs from the University of Chicago livers, and the green and red and blue here are just various definitions of replication, so they all give you the same pattern. Whereas you get closer to the start site, so this ranges from 200 kb upstream to 200 kb downstream. As you march closer to that start site, you get this very sharp, clean enrichment, suggesting that functional variation is enriched near promoters. So with that in mind, we'll return to this PRMT6 example, how we capitalized on this. So this is the Bayes factors from, now we're looking at the Seattle livers. And the gene itself is right there. So this is sort of highlighting how this enrichment near the gene is very pronounced. And if we zoom in here on this particular gene, you can see here's the extent of the transcript. Each of the SNPs that was tested for correlation with PRMT6 expression is indicated as correlation strength and base factors here on the y-axis. So you can see there's a number of SNPs here that all strongly correlate with expression. So what we were interested in was testing the hypothesis that there's a functional variant at the promoter that matters. So we resequenced the promoter for this gene. We did this for actually about 15 total genes, and I'll show some of the data for that. So indicated in star here, we sequenced about 2 kb upstream. We also sequenced the 3' UTRs. And essentially what this allows us to do is identify functional candidate variants. So here's an example from PRMT6. There's a SNP that's located about 150 base pairs upstream, and in fact, it's quite a bit closer depending on what gene model you use. So by default, we use REST-seq gene models, and it's 150 base pairs from that, but it's 50 bases up from a different model of the actual transcript annotation from Santa Cruz. And so here's the example of this individual SNP that was identified by resequencing. You have AA individuals have reduced relative expression compared to AT individuals, which are sort of in the middle, and then TT individuals have the highest expression level, so suggesting an additive effect here. And so what we're interested in was testing the hypothesis that this is a functional variant. And so this is where SwitchGear comes in. So the basic experiment here is we have a promoter of interest here, and it's hooked up to a luciferase reporter gene inside a plasmid vector. And then what we're going to do is, for each promoter, we've identified the different haplotypes that are present inside the liver samples that were analyzed. So we can clone each one of those variants and transfect them into cells. In this case, we did a HepG2 cell since it's a liver-derived cell line. And so what we're looking for is, in this case, we're studying haplotype 1, as we get some measurement of luminescence here that we infer that correlates with the strength of expression, which in turn should correlate with the strength of this promoter in vitro. And we can just march through and test multiple haplotypes, and hopefully we can identify a significant difference in this case here if we find that haplotype 2 here has a much stronger promoter and produces a brighter luminescence signal. Similarly, we can test for the effects of 3' UTRs, and this is, again, an assay that SwitchGear runs, where they have a basal promoter here, HSP70, with, again, luciferase supporter gene, and then we hook up the 3' UTR onto the back end of this luciferase gene, and the same experimental design where you're just testing each haplotype and comparing them to one another. So the overall experimental design here is for each gene that's associated with an EQTL, we resequence the promoter and we resequence the 3' UTR. And we identify common haplotypes here. So see that these are just different shades of colors here indicating different sequence variants associated at each one of these loci. SwitchGear then, so once we had identified the haplotypes, SwitchGear then cloned each one of these haplotypes into their luciferase vector system. And for each one of these haplotypes, they actually, it turned out to be important to clone them into multiple independent preparations of DNA. And each one of those preps gets transfected multiple times. So you have sort of multiple layers here of redundancy in the experiment to help minimize systematic noise. So the question then becomes, do the haplotypes that we sequenced and cloned and tested, do the luminescence data vary in the same direction as expression after you account for all those sources of noise? So this is one example where we got some very nice data. And again, getting back to the PRMT6. What I'm showing you here are these three sets of data points here come from the haplotype bearing the A allele at the PRMT6 -150 SNP. And each one of these columns corresponds to an independent mini-prep. Each mini-prep was tested 4 times. So effectively there are 12 measurements here clustered by the preparation of DNA. And this is in contrast to the T-bearing haplotype here. And what we find is that this is a significant difference between the T-bearing and the A-bearing haplotype that is reproducible both across mini-prep and across transfection replicates. And just as a reminder what the expression effect looks like, the AA homozygous individuals have reduced expression, the ATs are in the middle, and TTs are elevated. And this corresponds directly with the direction that we observe inside the luminescence data. strongly supporting the hypothesis that this variant is in fact functionally and causally relevant to the expression effect that we observe. Similarly, like I said, we tested 15 total genes with this paradigm and identified 2 other very clean or at least convincing links between expression and function via the luciferase assay. So here's an example of a promoter variant upstream of this gene called LBHC, where you can see the expression QTL measurement here of low expression of AA and intermediate of AC, and then presumably high of the CC individuals. And when we look here, the SwitchGear data are on the bottom part of this graph. And in this case, we actually had 4 distinct A-bearing haplotypes. So there are multiple variants often on these haplotypes, and it's not just the variants of interest. But in this case, you can see these are 3 different individuals of the most common A-bearing haplotype. And then here are 3 different haplotypes that, again, all bear these different shades of blue. The same format where each individual column corresponds to mini-prep and each individual circle corresponds to a transfection measurement. And you can notice that all the 4 A-bearing haplotypes all have relatively low expression compared to the C-bearing haplotype here, where we have 3 different mini-preps. You can sometimes see the variation from mini-prep to mini-prep, which underscores the need to do that in these kinds of experiments. But again, this strongly supports that individuals that carry a C at this particular variant have upregulated expression as a result of that variant upregulating promoter function. In this case, on the right-hand side, there's a gene called important 8, or IPO8, and we found the most strongly associated SNP after resequencing was found in the 3' UTR. And here is, again, this additive stepwise effect that you're observing for this GT SNP here at position 1311 within the UTR. And these data, the SwitchGear data in this case, the 3' UTR data tend to be noisier. But in this case, again, we find a statistically significant difference that's in the same direction as our EQTL effect that's observed in the actual livers. So here we're showing, again, multiple shades of blue indicating different haplotypes of the G-bearing allele, and multiple shades of red here indicating different haplotypes of the T-bearing allele. And like I said, it's definitely noisier, but this holds up to be both significant. And when we performed an independent analysis, which I'm not showing you here, but we SwitchGear actually re-prepped those UTRs for us and retested them. And in fact, the same signal held up as being highly significantly up in the T-bearing haplotypes and down in the G-bearing haplotypes. Again, consistent with the expression data. So in conclusion here, using this expression QTL mapping approach, similar to what other groups have also performed, we identify hundreds of strong and reproducible EQTLs in primary liver tissue. And there are thousands of weaker effects, but many of which are genuine. Many of these EQTL signals can be used to connect with GWAS results of complex phenotypes. So the example I mentioned earlier, sortilin-1 versus LDL, which also correlates with heart disease. This was actually found by a group led by Kathiristan in Boston, and the original expression association was identified using the Seattle livers that I talked about here. And this was actually followed up independently by this Musanugar, who also works with Kathiristan, in a really beautiful paper recently, where this turned out to be a long-range regulatory element. Using somewhat similar assays, this luciferase kind of model where they show functionally in vitro that they can identify the causative variant. Like I said, mentioned the VCOR connection with warfarin dose. And for the functional data here, of the 15 genes that we resequenced and SwitchGear generated the clones and tested for luciferase function, 3 of those 15 had highly significant functional effects that corresponded to the same direction as predicted by the expression measurement, suggesting that, in fact, a large fraction of EQTNs effectively can be identified just by focusing on a very small subset of the genome here, just looking at promoters and 3' UTRs, that we can predict by sequence and then test by functional assays like the ones SwitchGear runs. So with that, I'll finish up here. Just acknowledge Christopher Brown, who is a close collaborator at the University of Washington, all the individuals, at Chicago, all the individuals at UW, especially Mark Reeder and Debbie Nickerson, and then Nathan and Shelley at SwitchGear, who were instrumental in generating these data and helping with analysis, obviously, and then my funding sources and also funding for Mark and Debbie that supported a lot of this work. And with that, I will conclude. Thanks.
Okay, thanks very much, Greg, for an excellent presentation. So I just want to wrap up here at the end with a little discussion of some of the instrumentation that was used to generate some of the reporter assay data. So I want to show you just a few slides on the SpectraMax L Microplate Luminometer. And again, this is the instrument that was used to generate the reporter assay data that Greg just discussed. So this is an instrument that gives you highly sensitive luminescence detection, and we spec it out at about 0.2 femtograms of firefly luciferase per well, which would cover the vast majority of reporter-type assays that could be run. It has a very low background and crosstalk. It also offers the option for dual injectors if you need to do flash luminescence, and multi-detector configurations to get higher throughput in case you do need to screen through a number of different assay samples quickly. The PMTs are optimized for the typical wavelengths that you'll see in luminescence emission, including the reporter assay wavelengths that were shown in Greg's research, and also utilizes the ultra-fast photon counting technology to amplify the PMT signal and reduce noise, all of which gives you more reliable and reproducible data. The instrument's capable of handling a very wide dynamic range of samples, so if this is a concern in some of your assays, the instrument's able to handle it by using a number of settings, including an analog-only photon counting, which is ideal for very dim and medium signals. There's an auto range setting that covers an even broader range by combining the analog and photon counting abilities in the PMT, and also a max range setting, which adds an additional level of dynamic range to give you about 10 to the 9th orders of magnitude. So the luminometer is configurable. It's available with or without injectors. So without injectors, you're able to do glow luminescence assays. Configurations would include single-channel instruments with no injectors or 2 injectors, and a variety of other configurations going all the way up to 6 different channels or detectors and up to 12 injectors. So it's suitable for all different throughput levels that any user may encounter. And it is upgradable. So just to give you an idea of the higher throughput that adding on those additional detectors or PMTs would give you, if you were to do a typical luciferase reporter assay, this would be including injection and read time. To do the assay with a single detector instrument would take you about 16 minutes to read a 96 well plate, and so you could read about 15 plates in 4 hours. If you went all the way up to a 6-detector system, you would be able to read same 96 well plate in about 2.7 minutes, or read about 90 plates in 4 hours. So a considerable increase in throughput, and we also offer a SpectraMax plate handling system if you need additional automation capability. Okay, so just some of the other applications that can be run on the SpectraMax L. I mentioned the luciferase reporter assays. We can also handle cell viability assays, toxicity assays, monitoring for mycoplasma, ATP detection, a variety of luminescent ELISAs, and some GPCR assays, and a number of other assays in addition to those. And just to mention our software, the SoftMax Pro software is what operates the instruments. It performs data acquisition and full range of analysis and graphing features, and also offers up greater than 120 pre-configured protocols that allow the user to simply open a pre-configured protocol and run their assay with minimal setup time. Also, if customers require any GLP, GMP compliance, we offer software that can handle those needs as well. Okay, so with that, I want to wrap up, and if you have any questions for any of the panelists who presented today, please feel free to contact us. We have included our email addresses here, and thank you very much for your attention.